So our last speaker of the afternoon is Dr. Brendan Lanpher. We've heard so much about genetics, and I think we all can agree we all want to make more sense of it and get inside Dr. Lanpher's brain and know exactly what's going on. And so I've known Dr. Lanpher for a very long time. He and I are the only people in the room that probably know this until now, all of you, but he was my senior when I was an intern, so in pediatrics, and brand new green intern. And I always knew if I had a question, I could go to Dr. Lanpher, who would always know the answer, but at the same time be very calm. So, all right, Dr. Lanpher.
So thank you all for sticking with us till close to the end. I'm 100% biased, but I think genetics is the coolest thing ever. And so hopefully you guys will too soon enough. I hope to do two things with this talk that will sort of pull in opposite directions, but I think actually make some sense. One is to tell you that genetics is the coolest, most sci-fi, wildest thing ever, and also to make it make sense for you. So give me a minute and hopefully we'll get there together. So hopefully some of you have had the opportunity to do genetic testing for your condition. I know some of you haven't. I've talked to a few of you in the break. I think this is going to become more and more important, more than it is just interesting in the future, even than it is today. So why do we do genetic testing? We all want to know why these things happen, right? We don't want to just fix the problem. We want to understand it. And by understanding it, we'll have better tools to address the symptoms that these kinds of malformations have cause. As we understand the biology better, because there's lots of different ways you can end up with a vascular malformation, we'll be able to develop more targeted, more effective, less side effect-y therapies. And then these are genetic. Does that mean they run in families? Not necessarily. When people are thinking about having families of their own, are they wondering, is this going to happen again? Those are the kinds of questions we can answer if we know the genetic cause of someone's symptoms. So this is not for anybody to understand. But this is just to show the complexity of the genes we know of that can lead to vascular malformations. There are lots and lots of different genes that affect growth and tissue organization, which makes sense if you think about how complicated people are, right? It's not going to take one gene that just tells vasculature how to grow. But there's lots that work together in very delicate, coordinated ways. When I think about vascular malformations and genes, I sort of put them in buckets. One is the PIK3CA, AKT, mTOR signaling pathway, and we'll talk more about that. And the other big one is kind of the RAS MAP kinase signaling pathway. These are two different groups of genes that kind of work in sequence together that all end up affecting and organizing growth and differentiation of tissues. The one you've heard about most and is most relevant to most Klippel-Trenaunay patients is the PIK3CA mTOR signaling pathway. These are genes that affect the regulation of the cell cycle. So when cells divide, when they copy their DNA, how they do that, how they grow and proliferate, and when they stop proliferating is just as important as when you start growing. Most of the genetic changes that lead to vascular malformations lead to overgrowth or overactivity of these pathways. For most people who don't have vascular malformations, these genes are turned on and turned off appropriately. With these conditions, they get turned on and they don't get turned off or don't get turned off enough. And so the genetic changes typically are activating mutations. For the most part, not 100% of the time, but for the most part, disease-causing gene variations in these pathways are somatic, meaning they're present in the malformations. They're not something you were conceived with. They're not something that is present in every cell of your body. Again, there are exceptions to every rule, but that's a generalization with these genes. The other sort of big category that is more often different kinds of vascular malformations, syndromes, than sort of typical Klippel-Trenaunay are the RAS MAP kinase signaling pathway genes. Again, there's lots of different genes, but these are most commonly dominant conditions, meaning they can run in families. They often are more specific to just vasculature than the other genes in terms of the kinds of effects you can see. Now, let's talk about genetic testing. Genetic testing can mean a lot of different things to a lot of different people. What's most cool about this is how fast it changes. I graduated from medical school in 2001, started genetics in 2003. That was the year the Human Genome Project was completed. That was sequencing one person's genome. It took a team of about 20,000 scientists, 13 years and $5 billion to do it. I mean, I think about it like I think about the moon landing. I mean, it was just an incredible feat of scientific undertaking that, you know, nobody really thought they could do it in that much time. It was incredible. I saw a baby in our intensive care unit on Wednesday with a lot of health problems. We collected blood from that baby and his parents. We got a result from their genome this morning. So it can be done now incredibly quickly. We do it all the time in patients. It's usually not that fast. It's an emergency situation. But the technology change is just incredible. For people who don't know, this is Rosalind Franklin. She's the person who did the work that Francis Crick and James Watson took credit for and got the Nobel Prize for. What I always think is the coolest thing that any scientist has done, she looked at that picture and then just looked at the mix of carbons and nitrogens and phosphoruses in DNA and imputed the structure of DNA accurately. It's just a level of genius that we should all think about. This is just a sort of a history of kind of our understanding of genetics. We didn't really understand that DNA was what carried our traits and was passed from generation to generation until the beginning of the last century. So about 100 years ago was when we first realized that genetics was even really a thing and that DNA was important. Structure of DNA was first described in 1953. The first genetic disease that was actually, we understood the cause was Down syndrome. That was 1959. I'm not going to ask, but I suspect there's a lot of people in here that were alive before that. So this is really a new field of medicine. Gene sequencing any DNA at all, was really invented as a method in 1977. So I was alive then. My first summer job in the 1980s was sequencing DNA. I grew up right next to NIH. And you know, it took us 3 years to sequence 1,000 letters of DNA. Now that takes a second or two on our machines. Again, the Human Genome Project was started in 1990, finished in 2003. The current technology that we use for what's called next generation sequencing is really about 10 years old now. So this is really very, very new stuff and very exciting. What makes genetics cool and interesting, but also challenging, is that there's lots of different kinds of genetic variations, and there's lots of variations in all of us. So our DNA is packages of chromosomes. You can have a chromosome missing or extra. You can have sections of chromosomes missing. You can have single letters either missing or misspelled within our genes. And you need different kinds of chemistries or different kinds of approaches to look for each of those kinds of changes, at least in the best possible way. Again, the first human genome cost, you know, billions of dollars to do. Now it costs about $1,000, at least at the chemistry level. The interpretation takes longer. And so this has become much more accessible to far more patients, and is now starting to be covered by insurance. Thank goodness. That's a whole nother discussion. The most common test we do here when we're seeing patients with vascular malformation is whole exome sequencing. With that, we're looking at all the coding regions of essentially all the genes that a person has. This is, again, technology that's been around for a little over 10 years, maybe 12 years or so. And it detects all the currently known all the variations that we can find in the coding regions of genes. Right now, July 2022, the vast majority of the disease-causing variations occur in those coding regions. There's a caveat there. That's because that's the area that's been studied. In fact, the exome, or the coding regions of genes, is only about 1.5% of all of our DNA. The rest of our DNA has to be there for a reason. We're still learning what it all means. There's a lot of science yet to be done on this. Some of these areas are much harder to sequence, much harder to generate the data from because they're super repetitive, and the chemistry just gets challenging. Whole genome sequencing is kind of the newest kid on the block from a genetic testing standpoint. And this is where we sequence all the DNA, not just the coding regions of the genes. And so we are starting to move towards that, at least in some cases. There are some downsides to that for vascular malformation testing relative to exome sequencing, but for the most of the patients that I see with genetic testing, we're moving towards genome sequencing rather than exome sequencing. The differences in terms of yield of a diagnosis are pretty small for now. All right, what makes this hard is not really the chemistry anymore. Chemistry used to be the problem, it's now data. When we sequence any of us, average healthy person, if you look at their genome, has close to 5 million differences per person. That's what this 4.8 million nucleotides that are different in each person. Which makes sense. You look around the room, we're all different, right? This is a wonderful thing that we're all different in individuals, but it can be hard to tell which variations are causing issues and which aren't. Of those 5 million-ish variants, about 150,000 of which are rare enough that it makes us suspicious they could be related to disease. And we sort of filtered it. We have all these kinds of algorithms that filter down to try to understand which variations could be causing problems and which ones couldn't be. And it's still a work in progress, and that part is still quite manual. And that's where both the time and cost comes in, much more so now than the chemistry part. So what do we do here at Mayo? Is we ideally like to do whole genome or whole exome sequencing in what we call a quad. So that means 2 samples from the patient with a vascular malformation and a sample from each of their parents. We like, when it sort of works, to get a sample of quote unquote normal tissue from the patient. That's typically a blood sample or a cheek swab or something like that, as well as a sample of the DNA from the vascular malformation itself. Some people have very accessible vascular malformations. If you've got a big capillary malformation on the skin that is stable and not bleeding and not infected, a skin biopsy from that can be a great safe source of DNA. Some people have deep internal variant malformations. Some people have issues with skin integrity, and there can be other reasons where we can't get a sample safely. We'll just start with kind of germline testing or testing blood sample. We don't always have parent samples, but it's nice when we do because it can help us clarify, well, if this variant is found in both typical tissue as well as the affected tissue and is seen in a healthy parent, then it's probably not the genetic cause of disease. Sometimes we see 2 differences in one gene in somebody, and I can't tell if I'm looking just at your data itself, in most cases, but not in all cases. Are there 2 differences in one copy of the gene and none on the other, or are both copies of the gene abnormal? Remember, you get 1 copy of each gene from each parent. If I see 1 variant came from mom and 1 variant came from dad, then I know both copies are abnormal. If I see both came from 1 parent, then I know the copy from the other parent is normal. That's the ideal situation. We do this kind of testing and basically anybody we can, but not everybody has accessible parents. So we see kids who are adopted, we see all different kinds of situations. And so testing is available in other kinds of situations too. Sometimes insurance determines what we can and can't do, which is lovely. Before we do genetic testing, and I hope this happens at other centers, we like to go through a fairly detailed pre-test counseling. This is a big test. There's lots of stuff we can find that you might not be anticipating. First thing we talk about, well, we go through this whole informed consent form, but one of the things that I like to think about is secondary or incidental findings. The American College of Medical Genetics identified 73 different genes that are actionable enough, important enough, and dangerous enough if you have variants in them, that regardless of what the reason we're doing testing is, if you find something in those, we should tell you. That's one thing if you're my age and you're staring down the barrel of lots of age-related risks. But if you're a 5-year-old or a 2-week-old, do you want to know if you're going to get breast cancer or ovarian cancer and when you're well into adulthood, or do you want to know what your risk for colon cancer is? I tend to think knowledge is power, and that's a good thing, but not everybody's comfortable with that. And that's, it's something you can opt in or opt out of. Of course, everybody expects genetics to answer all the questions. I do too. It doesn't. I do lots of genetic testing on lots of medically complicated folks, not just with vascular malformations, and we reach a diagnosis or an answer between 30 and 40% of the time. That is far higher than it's ever been in my career. It's not nearly as high as I would like it to be, or as high as I think it should be. I think everything's genetic. Again, I'm biased, but I think as we learn more and can understand what these variations are doing more, we'll have more answers in the future. Of course, insurance and payment is a challenge. It's getting better. The first whole exome sequencing I ever ordered was in 2010. It cost the family $75,000 out of pocket. They were had plenty of means, and I was happy to help them. I wish it was accessible to everybody. Currently, if you're paying out of pocket, it costs a few thousand, not tens of thousands. And currently, we get genetic testing covered by insurance far more than we ever did in the past. It's still not 100% of the time. It's still a lot of hoop jumping typically. When we compare parents' samples to their children, we can find surprises. Sometimes we find that parents are related to one another. That can be an unexpected surprise. Sometimes we find that parents aren't related to their children, which is also an unexpected surprise. I tell people this is the slowest and most expensive paternity test you'll ever do. I tell every family every time beforehand, because I've been burned enough times that I don't want to ever be the source of that kind of information, but it does happen pretty frequently. And then finally, we talk about genetic discrimination. There is currently a law that was passed in 2008 that is called GINA. It's the Genetic Information Non-Discrimination Act, that says you cannot be discriminated against on the basis of employment or health insurance based on genetic testing results. And that's great. It's a good law. It could be changed. It hasn't been changed in whatever, it's been 14 years now. There were some efforts 3 or 4 years ago to try to weaken it. I think it should be strengthened. The same protection does not apply to life insurance or disability insurance or long-term care insurance. And so sometimes if we're doing testing for, it's not so relevant necessarily in Whipple-Ternowny syndrome or vascular malformation disorders, but if we're doing pre-symptomatic testing for someone with a family history of a neurodegenerative disease, we like to have those kinds of insurances in place beforehand. They can't take them away once you've got them, but they can deny them to you if you don't have them yet and you know about these kinds of genetic risks. It's a complicated thing. It's much more than checking a blood count or something in terms of what you might find. All right, you've had genetic testing. What do you do? What does it mean? I'm going to go through kind of what genetic testing results can look like and some of the nomenclature, the verbiage that can be a little opaque if you're not thinking about these all the time. So this is an example of a genetic test result from a patient we had last year sometime. And this is what it looks like. This is a positive result. Positive means they found something. Negative doesn't mean bad. It just means they didn't find something. And so positive, negative is the way we talk about these kinds of results. This patient had a causative variant in a disease associated with the phenotype, which was capillary malformations and hemihypertrophy. They did not find any variants that were possibly associated, so none of the kind of variants of uncertain significance. And we didn't find any of those secondary findings, those cancer risk type genes. This is what the variant was. It was a PIK3CA change, it was dominant. This is the specific variation. It was mosaic, de novo, and pathogenic. All right, what does all that mean? Well, let's talk about it. So the nomenclature, this is every time we see a genetic variation, it's going to have these kinds of symbols. And what does that mean? So C means its position. This was C2908. That's the position within the coding region of the gene. So C is coding. And so each gene has a start and a stop. And each letter along that has a different number assigned to it. So at position 2908, there's supposed to be a G. This was turned into an A. All DNA is A's, C's, T's, and G's, and it's supposed to be in the right order. But sometimes there are changes. And so this was a coding region location 2908 G to A. Sometimes, depending on the lab that's doing the reporting or depending on the type of variation that was found, instead of C, it'll be G, and it'll be genomic position. And so that's a position within the genome. And so that'll have a really big number, like a couple million or even a billion. because that'll be the position within the genome, and then it'll have the same kind of G to A type change. If it's in the mitochondrial DNA, mitochondria have their own DNA. It's different and separate from the DNA that's on our chromosomes and our nucleus of our cells. That'll start with an M. Mitochondrial variations are not really relevant to vascular malformation diseases, at least in a way that we currently understand, but it's included in a lot of the testing we do. And sometimes we find those variations unexpectedly, even when we're looking for something else. The same variation will be then translated into what it means to the protein. Most genes, not all genes, but most genes are more or less blueprints for proteins. Every 3 letters of DNA corresponds to 1 amino acid or 1 building block of proteins. And so for the most part, you can do the math, but don't. 2908 divided by 3 is approximately 970. And so at position 970 of the protein, switch an E to a K, which is the same thing as a glutamine to a lysine. So there are 20 different amino acids that have both a 3-letter code or a 1-letter code, just to confuse everybody. Sorry. Glutamine, the 3-letter code is nice, it's GLU. The 1-letter code for glutamine is E, I don't know why. Similarly, lysine, the 1-letter code is K. Again, I don't know why. But these mean the same thing. So this is this variation at the DNA level, the variation at the protein level. Sometimes there won't be a protein correlate. Sometimes the variation will be outside the coding region, but will affect the regulation of the gene, or sometimes it'll be a copy number variant or a splice variant. There's other kinds of variations that don't lead to a direct change in the protein code. So you might see just a DNA change. So there's lots of different kinds of variations. And then each variation is assigned a sort of relevance. Variations can be benign, likely benign. I don't know what it means, likely pathogenic or pathogenic. Most labs that do this kind of testing will only include the variants of uncertain significance, likely pathogenic or pathogenic variants on the report. Otherwise, the report would be thousands of pages long because we all have lots and lots of benign variations. But so you might see any of these kinds of assignments of variant relevance. Now, how do we come up with this? Don't read it. There's a very complicated kind of decision tree rubric that we go through to try to assign pathogenicity to a gene. It's simple if it's a common variation that we've seen a bunch before and know a lot about, and I've seen hundreds of patients with. But what if it's unique to you? We all have variations that are unique just in us. And we still want to have a way of predicting, is that likely to be disease causing or not? And we have tools, and it's not always perfect, but we're working on improving these all the time. Variations of uncertain significance are a common finding with genetic testing. The more we look at, the more we find. And again, it's not unusual for us to find variations that we can't quite say, is this really relevant or not? Some labs include these more than others. These can be reclassified with time. So it's not too unusual for us to get a call from a lab that, we did testing on 3-4 years ago. And they say, well, this variant we found in your patient, we've reclassified it. Most of the time, between 80 and 90% of the time, if something is a variant of uncertain significance, when it gets reclassified, it's reclassified as benign, because we've learned enough to say, this wasn't a thing to worry about. But 10 to 20% of the time, it's reclassified as pathogenic or likely pathogenic. So those are really variations we need to learn more about. Sometimes in a research setting, we can do additional testing here. We do RNA analysis. We can do animal models where we take a particular variant and put it in a mouse or a zebrafish or whatever, and to try to see, does it actually affect the organism? And that can help us predict if it's actually causing problems or not. All right, remember where it said autosomal dominant? What does that mean? Dominant diseases are genes where if you have one abnormality in one of the 2 copies, that's enough to cause problems. Autosomal means it's not one of the sex chromosomes. Most of us have either an X and a Y chromosome if we're males, or 2 X's if we're females. There are exceptions. Those are considered sex chromosomes, and the way those are inherited are different. But the autosomal means not a sex chromosome, and the dominant means if you have the disease, if you have one change in the gene, that's enough to cause problems. Recessive means both copies of the gene have to be abnormal to cause an issue. We're all carriers for a few recessive diseases. That's not unusual, but only if both parents pass on an abnormal copy is that relevant. All right. You'll see inheritance, and that can say maternal, meaning it was inherited from the mother, paternal, meaning it was inherited from the father, or de novo, meaning it wasn't seen in either parent sample. That could mean that it is unique to that patient, or it could mean that the parent has what we call gonadal mosaicism, which means they carry the mutation just in their ovaries producing eggs or testes producing sperm, which would mean that it's not detectable in them, but they could have multiple children with the same thing. That's a rare thing, but we always like to bring it up because no one will ever tell anybody the recurrence risk is 0 for something, because it can happen. The variant will be heterozygous, homozygous, hemizygous, or mosaic. What does that mean? Heterozygous means it's present in one of the 2 copies of the gene. It can be compound heterozygous, meaning you got 2 variations, each present in one of the 2 copies. Homozygous means the same variant is present in both copies of the gene. Hemizygous means you've got 1 copy of the gene and the variants in that gene, that copy. That's mostly X chromosome changes in males. Or it can be mosaic, which means the variant is present in some of the cells that were tested, but not all of them. So it's, you know, not at 50% of the DNA, it's at a much lower percentage. Germline versus mosaic. So some of the disease causing genes that lead to vascular malformations are dominant, meaning they can be passed from parent to child. And that means that variant is present in all the cells of the body. And that's what we call germline. That also means it's present in the ovaries and testes, so it can be passed on. Somatic mosaic means present in some of the cells. So I think about these in terms of when did the mutation happen? When did the change happen? With germline mutations, it typically occurs in sperm or egg development. It's present in every part of the person, and then it can be passed on. With somatic mutations, typically those conceptions occur without the mutation, but that at some point in early embryonic development, mutation happens. Those things happen by chance. There's nothing you can do to make them happen or prevent them from happening. We all have some somatic mutations. If they don't happen to be in the right spot of the right gene, they're not relevant for us. And so people with these kinds of somatic mutations, particularly if they're in these vascular malformation genes, will have areas of their body affected. You all can tell me about this far more than I can tell you. Where and when that mutation happens in what tissues will determine what the consequences are. If it's just, we've seen patients where it's just in one finger. We've seen patients where it's, a big portion of the body. And it very much depends on when and where that mutation happens. For people with somatic mosaic variants, those are very, very unlikely to be present in ovaries or testes, and therefore very unlikely to be passed on. The cells that ultimately become gonads or ovaries or testes are isolated within the embryo very early in development because their biology is so unique. And so it's very unlikely that these mutations would be present in those tissues. All right, this is more about germline versus somatic. With germline variants, sometimes we think about a second hit hypothesis. So you may have a mutation in every cell, but you see patchy changes. And that's because the gene may require a second variant to show up later on to cause problems. Usually with germline variations, you'd expect both sides of the body to be affected. So it's bilateral and it can be present all over. With somatic, you're looking for isolated, or one segment of the body that's affected. And again, it depends on when and where. With germline testing, any sample of DNA can do. So a blood sample, a cheek swab sample, spit in a cup kind of sample, anything will work. With somatic mutations, you got to look where the mutation is. So an affected tissue sample is going to give us the highest yield. Going back to the incidental findings, the 73 genes that are currently identified to be reportable no matter the cause, the indications for testing. These are mostly cancer risk genes. So the breast cancer risk genes, the colon cancer risk genes, some cardiology type stuff. Again, there's controversy about this in terms of, are we taking away autonomy if we're reporting this in a child? How actionable does it have to be included in this list? People go back and forth. Let's go back to this patient. Just this is the PIK3CA was the gene. This is dominant, meaning just one of the two copies needs to be abnormal to cause problems. We talked about what this variant means. It was mosaic, just as in this blurb down here. This was present in 15% of the reads from the skin and was not present in the patient's blood. And it was de novo, was not present in either parent. So that's today. Genetic testing is changing all the time. What are we going to be doing a year from now or 10 years from now? I don't know, but I have some ideas. Whole genome sequencing is the most comprehensive test we have currently. There are still some kinds of changes to DNA that we don't detect well. And so there's some newer sequencing chemistries that are probably interesting to me and no one else that will give us a better yield for those other kinds of genetic changes. We are still building up the databases to interpret what non-coding variations mean. Again, there are lots of them. They must have relevance. We don't know what they mean in a lot of cases. We are starting to explore doing transcriptome testing, which is looking at the RNA. So the RNA is kind of the middleman between DNA and protein. RNA is a lot harder to measure because it's kind of fleeting and the chemistry involved in isolating it and stabilizing it is tricky. But it can show us not just are the genes spelled right, but are they working right? Is that gene being expressed? Is that being transcribed into our RNA? So that's, I think, going to be really useful as they get better at sort of normalizing it as a treatment. Now, why do we do this again? This has been touched on by multiple other people, but there are treatment changes that can happen after genetic testing. All the blue stuff in here, maybe too small to read, are medications that target particular genes in these pathways. Right now, these are medications that target the normal copy of that gene, that sort of suppress the whole function of that gene. In the future, there's going to be other options. Gene therapy has been right around the corner for 25 years. I think it's still right around the corner. I don't think that this group of diseases will be the first targetable kind of system, but eventually I would like to get there. I am much more confident about or optimistic about what's called antisense oligonucleotide therapy, which is ASOs, where we can make molecules that target an individual's personal mutation. And instead of affecting the normal copy of the gene, we stop the abnormal copy of the gene being overactive. That's a really exciting kind of area of treatment development that I'm really involved with. That is, I think, shows a lot of promise. We'll see. But there's lots of other stuff coming too. And I think I'm just about out of time.